Introduction to Nanobiotechnology, Concepts, Opportunities, and Challenges, presented by Professor Natalia Kozan, Ivano-Frankivsk National Medical University. Objectives and Expected Outcomes. This lecture aims to cultivate a systematic understanding of nanomaterial application in modern diagnostics, empowering students to critically evaluate techniques. Fundamental concepts. Grasp basic nanobiotechnology terminology and concepts. Material science. Classify and understand nanomaterial synthesis methods. Ethical considerations. Recognize advantages, limitations, and ethical aspects. Diagnostic proficiency. Critically evaluate diagnostic techniques using nanomaterials. Defining nanobiotechnology. Nanobiotechnology is interdisciplinary field integrating biotechnology and nanoscience to create tools operating at the 1 to 100 nanometer scale. Biotechnology focus. Concerned with biological entities such as cells, proteins, and DNA, leveraging their functions for various applications. Nanotechnology realm. Engages with materials at molecular and atomic levels, enabling precise manipulation and novel properties. This unique intersection leads to innovations like nanobiosensors, nanoscale probes, and advanced targeted drug delivery platforms. Historical overview. From vision to reality. 1959. Feynman's vision. Richard Feynman introduces atomic manipulation in his seminal lecture, There is Plenty of Room at the Bottom. 1986. Drexler's molecular nanotechnology. Eric Drexler coins the term in Engines of Creation, envisioning molecular assemblers and self-replicating nanobots. 2000-2020. Rapid advancements. Significant developments in nanomedicine, biochips, CRISPR-based nanocarriers, and lab-on-a-chip systems emerge. Present day. Continued evolution from theoretical concepts to sophisticated real-world application in diagnostics and therapeutics. Key interdisciplinary foundations. Nanobiotechnology thrives on integrating diverse scientific and engineering disciplines. Physics. Principles governing optics, conductivity, and magnetism of nanostructures. Chemistry. Focus on reactivity and surface functionalization for precise interaction. Engineering. Design and development of advanced sensors and microfluidic systems. Biology. Understanding target molecules, proteins, DNA, and cellular interaction. This powerful synergy underpins the development of innovative diagnostic platforms. Terminological framework. A unified terminology is essential for clear scientific communication in nanobiotechnology. Nano-objects. Many structures falling within the 1 to 100 nanometer range exhibiting unique properties. Nanoparticles. Small entities, typically spherical or other defined shapes, widely used for various applications. Nanomaterials. Materials that inherently contain nanoscale components imparting novel characteristics. Bioconjugates. Nanoparticles precisely bound to specific biomolecules, such as antibodies or oligonucleotides for targeted action. Classification of nanostructures. Nanostructures are classified by their dimensions and specific functions, each dictating unique properties and applications. By dimension. One-dimensional, nanovires, nanotubes, for example, carbon nanotubes, silicon nanovirus. Two-dimensional, graphene nanosheets, for example, molybdenum disulfide, boron nitride nanosheets. Three-dimensional, nanogels liposomes. For example, monomeric nanogels for drug delivery, lipid-based nanoparticles. By function, diagnostic, sensors, biochips, for example, quantum dot-based sensors, lab-on-a-chip diagnostic devices. Therapeutic, drug nanocapsules, for example, liposomal doxorubicin, albumin-bound paclitaxel. Delivery. Gene therapy vectors, for example viral vectors, lipid nanoparticles for mRNA delivery. Synthesis methods of nanostructures. Nanostructures are created using two primary approaches, top-down and bottom-up, each with distinct advantages. Top-down approach starts from a bulk material which is progressively broken down to the nanoscale. This method includes lithography, patterning using light or electron beams, milling, mechanical reduction of particle size, sputtering, atomization of material in a plasma. Bottom-up approach involves self-assembly from individual atoms and molecules into larger nanostructures. Key methods are chemical vapor deposition, gas phase reaction to form thin films, biosynthesis, utilizing biological systems for nanoparticle creation, Sol-Gel technique, solution-based formation of colloidal particles. The bottom-up approach generally offers greater precision and superior functionalization capabilities for biomedical applications. Visualization and characterization techniques. Comprehensive characterization of nanomaterials requires a suite of advanced visualization and analytical techniques. TEM, transmission electron microscopy, provides ultra-high resolution structural imaging revealing internal morphology. AFM, atomic force microscopy, offers detailed 3D surface topography and mechanical property mapping at the nanoscale. DLS, dynamic light scattering, measures hydrodynamic particle size distribution in solutions, crucial for colloidal systems. NMR, nuclear magnetic resonance analyzes molecular structure, composition, and dynamics of nanomaterials. FTIR, Fourier transform in-framed spectroscopy, identifies chemical bonds and functional groups on nanoparticle surfaces. Critical evaluation of characterization methods. Selecting appropriate characterization techniques is vital for robust analysis and nanobiotechnology research. TEM and AFM are indispensable for direct structural visualization, providing critical insights into morphology and surface features at high resolution. DLS is particularly fast and accurate for assessing particle size and aggregation in colloidal systems, making it suitable for routine analysis. FTIR and NMR, while less sensitive to the precise nanoscale dimension, are profoundly important for understanding the chemical composition and surface functionalization of nanomaterials. Modern nanobiotechnology research commonly integrates TEM plus DLS plus spectroscopy for a comprehensive and robust analysis of nanomaterials, ensuring accuracy and reliability. Nanobiotechnology, revolutionizing science and industry. This presentation explores the transformative impact of nanobiotechnology across various sectors, highlighting its application from targeted drug delivery to environmental remediation. We will delve into key innovations and future prospects. Overview: Mapping nanobiotechnology applications. Medicine and pharmacy. Diagnostics, therapeutics, monitoring. Food industry. Smart packaging, freshness sensors. Agriculture, nutrient delivery, pest control. Environmental science, water purification, soil remediation. Nanobiotechnology's broad spectrum of applications is rapidly advancing, bridging critical gaps in healthcare, environmental sustainability, and food safety. Medicine, nanoparticles for targeted drug delivery. Liposomes, biocompatible vesicles for hydrophilic or hydrophobic drugs, for example Dexil. Dendrimers, hyperbranch structures with multiple functional loading sites. Micelles, polymer-based self-assembled structures enhancing bioavailability. These nanocarriers enable precise drug delivery, reducing toxicity, and enhancing therapeutic efficacy. Nanorobots, precision in surgery and diagnostics. Theoretical models, development of nanobots and nanorovers for intricate tasks, vascular surgery, magnetically guided micro or nanoscale objects for thrombolysis, intracellular probes, nanoprobes for high-resolution visualization. Challenges: ensuring biocompatibility and achieving real-time navigation remain key hurdles for clinical translation. Biosensors and nanochips, advanced disease diagnostics. Biosensor types: electrochemical, optical, plasmonic. Application: glucose, pathogen detection, protein markers, DNA, cancer, exosomes, viruses. Highly sensitive biosensors, including electrochemical, optical, and plasmonic types, are revolutionizing early disease detection. Genetic engineering. CRISPR-linked nanostructures. The CRISPR-Ca9 system offers precise gene editing, but delivery remains a challenge. Nanostructures provide solutions. Lipid nanoparticles efficiently deliver Cas9 plus cgRNA into cells. Gold carriers, stable and versatile platforms for gene cargo. Polymeric nanogels, biodegradable systems for controlled release. Example: successful correction of beta thalassemia mutation using nanoparticle delivery. Agriculture: smart delivery for fertilizers and pesticides. Nanotechnology enhances agricultural efficiency and sustainability. Nanocapsules: controlled release of nutrients and active ingredients, reducing waste. Nanopesticides: lower dosage, reduced environmental runoff, and precise pest targeting. Examples: agri-nano-zinc for enhanced zinc uptake, and nano-argentum formulations for antimicrobial protection. Environmental protection: nanomaterials for remediation. Nanoscale zero-valent iron, effective in removing heavy metals and nitrates from contaminated water and soil. Nanocomposites, high-efficiency absorbents for oil spills and various pesticides. These materials offer high surface area and catalytic activity, making them superior for environmental cleanup. Food industry: nanosafe packaging and freshness sensors. Nanotechnology enhances food safety and extends shelf life. Antimicrobial nanofilms, using silver and zinc oxide nanoparticles to inhibit microbial growth. Gas sensors detect ethylene and ammonia for real-time freshness monitoring. Example: smart packaging for fresh meat with embedded temperature and gas sensors, ensuring optimal quality for farm to table. Nanotherapeutics, advancements, challenges, and future. Limitations and clinical efficacy. Regulatory barriers, navigating EMA and FDA approvals. Material issues, addressing biodegradability, toxicity, and immune response. Clinical trials, phases 1 to 3, with a need for population diversity. Future trend: Europe Nanomedicine Roadmap 2025-2030. Deep personalization, tailored therapeutic approaches. Nano AI platforms, integrated artificial intelligence in nanomedicine. Organ on a chip, integration with advanced biological systems. Scaling and cost challenges. Mass production, difficulty scaling lab technologies for large-scale manufacturing. Synthesis cost, high cost of producing single-molecule sensors. Case study, comparing carbon nanotubes with polymer analogs. Nanotechnologies in wartime, biothreat detection, biosensors for rapid identification of biological threats. Personalized first aid, 3D printed nanodrugs for customized emergency care. Military biotech, leveraging experience from nations like Israel and the USA. Discussion: is nano a panacea? Reassessing expectations, high versus real-world outcomes. Field startups and clinical trials. Nanotechnology as a tool, not a cure-all. Section 3. Risks and limitations. This section will cover the potential risks and limitations associated with nanotherapeutics and nanomaterials. Toxicological aspect of nanomaterials. Nanotoxicology. Definition. Studies biological effects on nanoparticles in living organisms. Nanosize impact. Enables membrane penetration, tissue accumulation, and oxidative stress. Test systems. In vitro, cell lines. In vivo, animals, and organ-on-chip models. Example, toxicity of carbon nanotubes in lung tissue. Nanomaterials in the body. Entry routes, inhalation, dermal, oral, and intravenous exposure. Biopersistence, some nanoparticles are non-biodegradable. Carcinogenic potential, depends on shape, surface, modification, and dose. Environmental hazards. Sources: nanocosmetics, nanodrugs, and pharmaceutical effluents. Potential effects: inhibited photosynthesis in algae, altered soil microbiota. Bioaccumulation, accumulation across various food chains. Ethical and regulatory landscape of nanotechnology. Exploring the complex ethical dilemmas and regulatory challenges in the rapidly advancing field of nanotechnology. Ethical dilemmas. Molecular manipulation, genome editing, ethical boundaries of altering embryonic genomes and influencing cognition, nanorobot use, future concerns and consensus on nanorobots in neurotechnology. Informed consent in nanotherapeutics. Patients in clinical trials must fully understand nanoformulation contents, mechanism of action, associated risks. Ethical obligations include accurate translation and mental capacity assessment. Access disparity, global north and south. Technological inequality, US and EU lead. Many African and Asian countries depend on imports. Nanoelitism risk, nanomedicine potentially accessible only to wealthy regions. Equitable access, need for policies ensuring fair global access to nanotechnologies. International regulatory documents. OECD, Nanomaterial Safety Testing Guidelines. ISO, Biocompatibility 10993, Risk Management. TR 12885. FDA, Nanotechnology Task Force Guidelines. EMA, Evaluation Framework for Nanodrugs. Lack of harmonized legal framework. EU regulations. REACH, CLP, Nanorisk Framework. US protocols. Different safety and approval process. Ukraine. Partial mention in pharmaceutical dosages. Urgent need for global nanotechnology policy and common classification standards. Conclusions and discussion. This section summarizes key achievements and challenges and poses discussion questions for further exploration. Summary. Achievements and challenges. Potential. Nanobiotechnology offers major potential in medicine, agriculture, and environmental science. Key issues: toxicity, ethics, public trust, legal regulation. Central goal: safe, effective, and ethical integration of nanotechnologies into practice. Discussion questions. 1. Ethical nanomedicine. Is ethical nanomedicine truly possible? 2. Innovation versus safety. How can we balance innovation with safety? 3. Healthcare readiness. Is our healthcare system ready for these technologies? Self-assessment and homework. Self-assessment questions. Types of nanoparticles for drug delivery. Liposomes versus micelles. Regulatory agencies overseeing nanotherapeutics. Ethical dilemma. CRISPR and nanotechnology. Importance of nanomaterial environmental impact assessment. Suggested essay topics. Nanoparticles in infectious disease diagnosis. Biosensors based on nanomaterials. Comparative analysis. Social risks and inequalities in nanomedicine access. Nanotoxicology. Modern safety evaluation approaches.